Hello, everyone, and thank you for coming today. I'm Erika, and before to get started, I would like to give you a couple of information. Firstly, this webinar is recorded, and the recording will be available on biostimulant.com website and on our social media in the next days. Secondly, we invite you to use the Q&A box that you can find at the bottom of your screen to submit the questions. We will collect your questions and answer to at least some of them at the end of the presentation. The topic of today is metabolomics, a systematic approach for the development of highly efficient plant biostimulants. It's my pleasure to announce our speaker, Paolo Bonini, founder and CEO at OLOBION, a laboratory of metabolomics analysis located in Barcelona, Spain. Welcome, Paolo, and the floor is yours. You just have to share the presentation.
Thank you very much, Erika. Thank you, everybody, for attending this presentation. I will start sharing my screen. I have a little PowerPoint I have prepared for today. So today we are talking about metabolomics and how metabolomics can help the development of new plant biostimulants. Before starting, I want to introduce a little bit about what is metabolomics. So metabolites are the end product of cellular regulatory process, and their levels can be regarded as the ultimate response of biological system to genetic or environmental changes. Metabolites are very important because are the end product of everything. So as there is a transcriptome and a proteome that is more studied, there is a set of metabolites that is called metabolome. This definition has been done by Oliver Finn in 2002. So why metabolomics in plant biostimulant is important? We will talk about this during my presentation, and I want to show you this picture about a strawberry field, treated and untreated. Maybe you cannot see the difference in phenotype in this case, but can be a real difference in the metabolome of the plant. So metabolomics can be important not just to assess why the phenotype is changing, but also for the quality of the plants and the fruits. So what is happening behind the scenes? A little bit of numbers about biostimulants and metabolomics. It's a field that is developed in the last 5 years from 2015. And there is few papers about this, like 1,600 papers about biostimulant and metabolomics. The most important journals you can see here in the presentation. So who is doing metabolomics and biostimulants? So the top 100 researchers about this field are the same that are working in the biostimulant group. So we have Professor Giuseppe Colla, Professor Rufel Youssef, and Luigi Lucini, like the most important people developing this field in the moment. And this is a map about the co-authors. So the same color, it's the same group. And is mostly developed in Italy, even if now is becoming more important also in Canada and South Africa. So metabolomics is about a flux. First, we have the genome. So the genome contains all the information about an organism. Then this genome have to be translated into the transcriptome. So we have RNA that code for proteins. So we have the proteome that generate the metabolites. The metabolites can be inside our cells or outside, that is the exposome, the metabolites that we are exposed to, chemicals. Also biostimulant can be the exposome of a plant. And this is a wheel that move also the proteome and the transcriptome, so are interconnected together. So it's not just the end product of everything, is also able to influence everything. Also, the metabolome change in the time. So this is a figure from a paper that in each study of the plant, we have different type of metabolites that have been expressed. This is limited to the plant hormone, but we can see like gibberellin and auxin have been expressed in different growth period. So the metabolomes is important to know that is changed with the time. So this is the paper I took the figure. And also a very recent paper in the New Phytologist talk about the matter flow and the phytochemical connection in the food web. So we know that the plant are the basis for food production, but the phytochemicals in the plant have an effect in all the food chain. So have an effect on the soil, microbials that live in the soil, have an effect on the animals, have an effect on the insects, and then have an effect on the humans. So all these are important. The metabolome is important and have a flow directly in all the living organisms. The approach we use for metabolomics is simple, but also very complicated. So we start with a trial when you have a control plants and treated plants, one without plant biostimulant, one with plant biostimulant. You do the trials, you can sample your plants in different time if you are interested in how the metabolomes change in time. You have to do a sample preparation. The sample preparation will change depending of what you are interested in. If you are interested more in polar metabolites or in lipids or in hormones, you can choose an extraction type that is focused on the subset of the metabolites you are interested in. So then you have untarget approach or target approach. The untarget is try to have the maximum number of annotation inside your sample. And the target approach, for example, if you are just interested in amino acid composition of the plant, so you focus on that. You are more specific, you can quantify, while in the untarget approach, you try to reach everything, everything you can. And then there is a huge work on bioinformatics when you have to annotate the molecules and you also to interpret the data. That work is very hard also for pathway analysis and statistical interpretation. So let's talk about the technology that is behind the metabolomics. In our cases here in OLOBION, we have the last generation of instrument is an Agilent 6550 with ion mobility. We use for both metabolomics and lipidomics application. Lipidomics is a subset of metabolomics that focus on lipids. So we use a new developed technique for identification we called 4DID, four-dimensional identification. So when you do metabolomics, you have signals inside your mass spec. So these signals you have to translate in annotation. And for translating in annotation, we use 4 dimensions: the mass charge, the retention time, the drift time, and the fragmentation spectra. So I will explain to you what there is behind this analytical chemistry part. So the first things you do inside this machine is weight your molecule. So there is the mass charge you have. No? So for example, you have a molecule that weighs 490 Daltons, but you don't know exactly what is it, because just with the weight, you can narrow down the search of maybe 100 different molecules that can be in the plants. So with just the mass, this figure is very explicative, because with the mass only, you doesn't know what can be butter, can be whatever behind. No, in this case, it's chocolate, but you cannot know just for the mass. The other thing that help the identification is retention time. Inside the HPLC, we have a column. This column is full with silica particle with a specific coating. In this case, is a C18 coating, a carbon with a carbon change with 18 carbons. So in this case, the molecules are retained differently for different chemistry. So the behavior of retention time are molecules that, for example, in C18 elute early, that means it's very polar. And if it elute at the last part of the chromatogram, that means it's more lipids-like. So this behavior help us in the identification. We published recently in the past year a paper about retention time prediction with a collaboration with the Riken Institute and also UC Davis with Oliver Finn group about how we can predict this time and apply to compound identification. And last is the drift time. The drift time is something is very new in the metabolomics, and is about how the molecules behave in a gas. So the point is you have a tube filled of gas that is the drift tube. So these are the animation, the molecules that flow in that, the gas molecules in the flow. So you trap your molecules that these molecules, if you can see, have the same, have different structure, but the same formula. But having this different structure behave differently in the gas. So if we release, you can see that the two molecules have different drift time. So that helps also in the identification of the molecules. So we have talked about the analytical process of metabolomics. We talk about what metabolomics is, and now we focus on the application of metabolomics in plant biostimulant. One of the most important application is understanding the mode of action of plant biostimulant. No? So the mode of action, you have a phenotype that your plant biostimulant can modify. You want to define the target you want to you focus. For example, you want to focus your trial on salinity or cold stress or heat stress. So you have to plan your agronomical trials with your plant biostimulant and with your desired results. Your product will be changing the biochemical part of the plant. So we will change small molecules concentration. And that is because it's changing biochemical pathways. So the plant is sensitized more a particular compound than the other. Also, there is a changing in gene expression that can be measured. But in our case, we focus on two omics strategy, that is phenomics and metabolomics, because they integrate very well. From one side, you have a specific field trials with cameras that can give you a lot of agronomics results, a lot of data that can be integrated with the metabolomics data that show you what is happening at biochemical level for that particular phenotype. This is a picture about a phenomic platform that is developed by Arcadia in the University of Latouche, where Professor Colla is doing this. So this is a specific trials that we have done about the developing of new plant biostimulant for strawberry. So we have this customer that have 3 different protein hydrolysate. We have done the test in the field, and the 3 plant biostimulants stimulate the number of fruits per plant and the total yield. So we have, if you just base your analysis on agronomical data, you say, okay, the 3 biostimulant works very well. They stimulate growth, they stimulate production, perfect. But in the case of strawberry and the case of all agronomical or plants product, we want to know also, what about the quality? Does this product change also the quality, or is the same for the 3? No? Using metabolomics, you can answer this question, not just about the Brix grade. That the Brix grade is very easy to measure. When you want to have the whole picture, no? What about the defense of the plant? What about, is it able to resist a cold stress in the last of the season? So we can answer this with the metabolomics. And we have done, this graph are the synthesis of the whole metabolome. So we align all the annotation of metabolites inside the pathway of that specific plant. So we group these metabolites in pathways. So for example, this you have anthocyanin biosynthesis that group all the metabolites we have annotated and measured inside this family. Then you have flavonoid biosynthesis, starch and sucrose metabolism. So you group these metabolites in these families. And this picture is the dots is the fold change. The biggest is the dots, the most important is the changes. And blue, that means that it is downregulated, where red is upregulated. So here we see that the first plant biostimulant downregulated flavonoid biosynthesis that are very important for human health, are antioxidants. Also for plant are important for defenses, for example. So you have riboflavin that go down, that is a vitamin. You have the starch that is in the middle, a little bit higher, but not significant. So this kind of biostimulant, okay, we know that increase the yield, but do not increase quality of the plant. Let's see the second one. The second one increase a lot the flavonoids, so it's good, it's plant health, but the starch and sucrose metabolism go down. That means that the fruit can be more healthy for human, but is not so tasty because have not so much sugar in it. So it's kind of you're in the middle. Okay, you know that the fruit is, the yield is higher, compounds that are important for human health are higher, but still you miss something. The third parent biostimulant was the chosen one. Why? Because it stimulates everything that was important. It stimulates starch and sucrose metabolism, so you go up with the sugars, and you also go up with flavonoids. So, and the anthocyanins. So anthocyanins are important for colors, and flavonoids are important for human health, as I told you. So these kind of results can help to choose between different biostimulants that phenotype level are exactly the same, but metabolic speaking are different. This is another example that we have published in Frontiers of Plant Science, is how metabolomics help to evaluate plant biostimulant in pepper. So we had a mix of trichoderma and mycorrhiza applied to a pepper field that was actually in production, was in Almeria. So we analyze how this product changed actually the metabolome of the plant. And was doing also, we have done also in 2 different times. So this is the publication, you can see. These are the data from the field, the agronomical data. So we can see different fruit collection. In all of them, the biostimulant increase the fruit yield, the fruit number, and we want to see the same. What happens to the plant? Are the plant behaving better? And in what kind of metabolites? So we can see here at 43 days after the transplant and 131 days after the transplant. So are 2 different timing. You can see how this changing in the metabolome. This is also all the metabolites grouped for chemistry family. So in the other case was pathway, this was chemical family. So we can see that, for example, carotenoids are increased, also saponins that are important for plant defense. This was leaves and phosphatic acids. So all of that, them in the paper, we disclose every single family class, and we say this is important for cold stress, this is important for plant defense or for plant growth. No? So we can see that you are stimulating not just the production, but also the important things in the plant that are defense, are also the quality of the fruits. This is another type of graph that we have done for the publication, is a network that correlate all the chemistry family. So for example, you can see the red dots are upregulated, the blue dots are downregulated. The part of flavonoids and coumarin and lignans that are antioxidant are always upregulated. We have auxin and cytokinins that also are upregulated. That makes sense because we see that we have an improvement in the production. And then you see that the graph changes between time after 90 days, 3 months. You have the plants. The first plant was without fruit. No? As you can see in the plant, there is no fruits. And after 90 days, there was fruit. So here you can see more carotenoids and vitamin A that are stimulated in the production on the leaves and then go to the fruits. Also, metabolomics can be important in bioactive compound discovery. So there is a lot of known plant hormones that are known from several times. One example is indolacetic acid, AIA, and this is very known that influence the plant growth, but is not known what else is also contributing in the plant growth. So this work that we have published in Frontier in Microbiology, we have developed a metabolomic procedures to understand better what kind of metabolites also help this function of indolacetic acid. And also we elucidate the pathway of production of EIA of this particular strain of bacteria. These are the results from the paper. One is the pathway that we know that bacteria produce indolacetic acid with this pathway. And these are all the metabolites that are inside exudate from the bacteria and inside the broths that we use for plant treatment. So there is a huge amount of compounds that are also there. And what metabolomics can do is also help you to find out which one is active together with the indolacetic acid. This is a work that we are still developing. Also, I want to give you some perspective. One of the things that can be important for the field in the next year is the combination of metabolomics, phenomics, and machine learning. Why? Because metabolomics and phenomics give you a huge amount of data. We're talking about several gigabytes of data that are related with one single experiment. So the point is how you relate everything. So this can be also important for discovering new pathway, for discovery new ways to stimulate plant growth. So this can be done with machine learning. We are working on it or integrating all this database. And this is the perspective I want to share with you in this presentation. So thank you very much for attending. Here there is my contacts. I am also in Twitter, Paolo Bonini 6. And now I am open to your question.
Well, thank you very much, Paolo, for this interesting presentation. And we have seen how metabolomics can help the development of highly efficient plant biostimulants and also deeply investigate the mode of action of the products. Now we will have a Q&A section. I just wanted to remind to all of you that it's still possible to submit the questions using the Q&A box. We have already received some questions, so I'll begin immediately with the first one. If we are interested in develop plant biostimulants that increase plant growth, can we focus in just a subset of metabolites?
Yes, good question. Yes, it's what I was speaking in the beginning. There is two approaches. One is on target and one is target. So if you want to focus just in how your product influence plant growth, we can focus on only in plant hormones, for example. There is a database of plant hormones that is 250 different compounds. We can focus on that, find that in the plants, and extract precisely. So we can give an interpretation based on just a subset of metabolites that are known to be related to plant growth.
Okay, thank you. We have another question, which is, can metabolomics helps to detect plant abiotic and biotic stress?
Yes. So one of the things that the biostimulant are known of is protecting the plant from cold or heat stress or water stress. So you can design an experiment where you treat the plant with that threat, and then you treat with plant biostimulant, and then you find out what kind of pathway or metabolites are stimulated or downregulated in your plants. So the answer is yes. Also for phytosanitary product can be very interesting, the application of metabolomics, because you can know exactly how work your phytosanitary product. It can be natural or microbial. So I think metabolomics can give a lot to the agricultural development.
Okay, thank you. I take just a moment to answer to one question, which is about your presentation. Someone is asking if we will provide this presentation, and we want to provide the presentation, but the recording of this webinar will be available next week on biostimulant.com website and on our social media. So all of you will be able to see it again in the next days. Then coming back to the questions for you, Paolo, there is another one who says, which are the main limitations nowadays that hamper the wider diffusion of metabolomics in biostimulant sector? And I will also add another question about the cost. So it common here that metabolomics is very expensive. So if you want to discuss these both questions together.
Yes. So the question about the diffusion of metabolomics, I think metabolomics is the less well-known technique, because you can see more paper about transcriptomics with the plant biostimulant. There is a lot of also qPCR, but metabolomics is quite new in the field of plant biostimulant. So I think that nowadays can be applied wider. The costs are not very expensive. In our website, olobion.ai, you can have, there is a list of prices. It depends if you are more interested in primary metabolism or secondary metabolism. The techniques are different. There is gas chromatography or liquid chromatography. So a study with like 2 treatments, 10 sample with plant biostimulant, 10 sample without plant stimulant, we are talking about 2,000, 3,000 euro. So it's not that expensive. Then you have to add also the cost of the agronomical trial, but at least it's not expensive. It can be like a qPCR or a transcriptomic analysis. It's not so different, but it's not so common to find out. So that's why we opened the lab to give the possibility to everyone to do this kind of analysis.
Okay, thank you. We just have another interesting question about what we have seen in your presentation about all the data we obtain through metabolomics. So we have, for example, thousands of compounds and so data to manage. Maybe we can get lost. Do you have in your company a service for analyzing this data and so make them more understandable?
Yes, we focus a lot on that, on the interpretation of the data. We have a department here in the company of bioinformatics that his focus is just in identification of compounds. That is a critical step. It's very difficult step, so you have to really focus on that. And then pathway interpretation. So you have this annotation and you want to know what kind of influence these have in the plants. So we group for pathway, we group for chemical families. The graphic I showed to you, you need to understand better what is happening, because if I give you a list of 200 or 500 metabolites with no explication at all, it's impossible to know. So this kind of graph grouping for pathway or chemical are important for interpretation, because we want to give insight, not just metabolites. So it's our business is that.
Okay, and so to go a little more deep about metabolomics, someone asked, metabolomics can be addressed to primary or secondary metabolites of plants. Based on your experience, what do you suggest as first approach in understanding the biostimulant activity of a product? So analyzing the primary or secondary metabolites.
Okay, it's depending of what you see in the phenotype. So, for example, here we do both, no? And normally we try to do both. But if you are, for example, interested in more plant defense stimulation, if you are interested in antioxidant secondary metabolites that influence the cold and hot stress, so go for liquid chromatography and secondary metabolism. If you're more interested in the plant growth, most of hormones, you can see very good in GC analysis. So I will go for that. If you are interested, for example, in root exudate, so you have to do with the GC. The deal is finding a lab that have both technique and can help you to decide before you start the experiment what you should do. Because there is also lipidomics, for example, that focus just on lipids that are very, very interesting, and it's a new field that in plant biostimulant will came through.
Okay, thank you very much, Paolo, for your answers. It was the last question for today. I would like to thank you again for the insightful and very useful presentation. And I would like also to thank everybody for attending the webinar. If you still have questions, don't hesitate to submit them on biostimulant.com website, and our scientific committee will get back to you with the answers. I would like also to remind you that the recorded version of this webinar will be available soon on biostimulant.com website and also on our social media. Have a nice day, everybody, and we hope to see you again on the next webinar.
Thank you very much. Thank you very much. Bye.